Chromosome and DNA Mutations by Leslie Adams, Nick Knight, and Madison Mann. A mutation occurs when the structure of a gene has been changed in some way so as to result in a variant form of the gene. There are two general types of mutations, the first of which is chromosomal mutations. These include mutations in which the number and/or structure of chromosomes has been altered. The second type are point mutations, and these occur when a single gene has been changed at the molecular level. For example, in the image, a single nucleotide has been changed from T to C. There are four basic types of chromosome mutations, with the first example being a deletion. In deletion mutations, one or more nucleotide bases are absent or lost from the DNA sequence. An example of this would be Kreutzfeldt syndrome, where there is a deletion on chromosome 5, causing a cat-like cry and severe intellectual disabilities in those affected. The next mutation is duplication, where a section of a specific chromosome or genetic material is replicated. An example of this is Fragile X syndrome, where there are duplications on the X chromosome causing cognitive impairments. Third is a translocation mutation that is characterized by part of the chromosome being relocated. One example of this is acute myelogenous leukemia, where part of chromosome 22 has moved to chromosome 9, causing an excessive growth of abnormal cells that builds up in the blood and bone marrow. Lastly, an inversion is a change in genetic material occurring when a portion of DNA breaks off and attaches itself in a reverse order. An example of such is hemophilia, a bleeding disorder characterized by an inability for blood to clot appropriately and causing excessive bleeding and joint pain and swelling, among other things. The attached image gives a brief visual of how exactly these four mutations occur. As for point mutations, there are three general types, the first of which are silent mutations. These occur when a codon is altered but still results in the production of the same amino acid. There is one silent mutation known that increases people's risk for developing cardiomyopathy. The second type is missense, in which a codon is altered and results in the production of a different amino acid than the wild type. One example of this would be sickle cell disease, in which glutamic acid is replaced by valine and causes patients to have many sickle-shaped red blood cells, resulting in severe pain. Lastly, a nonsense mutation occurs when a codon that normally codes for an amino acid is instead converted to one that provides a stop codon. An example of such would be cystic fibrosis, a condition where patients experience their lungs and digestive systems clogged with excessive mucus. All of these mutations can occur in two different types of cells, the first of which is termed somatic. These are non-reproductive cells, and in these types of mutations, clone cells will be created through mitosis that will then all contain the mutant gene. As for germline mutations, these occur in cells that will eventually give rise to gametes. Through meiosis and sexual reproduction, the mutant gene will be passed to approximately half of the patient's offspring. This concludes our video for chromosome mutations. Thanks for watching.